My pleasure to introduce President and Chief Executive Officer of Lion Biotechnologies, Elma Hawkins, who I think has some slides prepared to review the TIL platform and the exciting things going on in this cancer immunotherapy field.
Thank you, Josh, and thank you very much for the opportunity to represent Lion here this afternoon. Just please do a little bit of speed reading here and take a look at our forward-looking statements. I thought it would be important just to remind everyone again as to why TIL are so important in solid tumors. First of all, there is a very large database of TIL experience with over 600 patients worldwide now. And these are, to a large degree, phase 2 clinical studies. TIL recognize neoantigens, and the neoantigens are unique to tumor cells. And so because of this, we have a less or a very, very small chance of on-target but off-tissue adverse events. Remember that every patient's tumor is antigenically distinct from another. And so by making a TIL cocktail, essentially, for a patient, you're doing it for that patient and because you are targeting those particular antigens. The other huge advantage is that it's one treatment and you're done. And I will talk a little bit later about the how easy it really is to prepare TIL, and we can now prepare it with 90% plus success rate, certainly as reported by the NCI and Moffitt. And then on the right-hand side there, you'll see the little diagram that we take tumor from the patient, we expand it, and reinfuse it into the patient post lymphodepletion, and follow that with IL-2. Some of the data that was recently reported by the NCI can be seen here. This is the largest single study that had been conducted with TIL in metastatic melanoma patients. These patients have all been very heavily pretreated with chemotherapeutic agents, but for the most part, biological agents, IL-2, interferon, ipilimumab, and also pembrolizumab and nivolumab. So the overall response rate observed in the study was 54%, and the complete remission rate was 24%. This is certainly very impressive data that in many ways is unparalleled. Especially if you look on the right-hand side to see the Kaplan-Meier curves there. And the two curves represent really the lymphodepletion with or without total body irradiation. The outcome was that whether you add total body irradiation or not, it really does not make any difference. And so from a patient perspective, this is very good news, right? Because patients don't have to be subjected to something this toxic. But you can see very, very nice survival here. And in an older study, we have patients now who are out to about 11 and 12 years. They're still free of disease. So those really do present functional cures. And I just want to review this as a snapshot of all the clinical trials that are relevant. So LN-144 is our phase 2 study. And then in green, you have two studies by the NCI. The one is what I just discussed. And the other is an exciting study where we're in the era of combination therapy. And so this is to combine TIL with pembrolizumab. There's a study at Moffitt that just recently has gotten IRB approval, and that is to study TIL with nivolumab in frontline patients. So that is going to be quite interesting. Then we're also conducting studies in other indications, most notably cervical cancer and various types of head and neck cancer. And then in preclinical research in the lab, we're currently working on breast, lung, and bladder cancers. And that, of course, is very exciting to us because we can definitely grow TIL in these indications. It is not as difficult as people have expected. And so these can go into clinical trials certainly by next year. So just to recap very quickly, we have LN-144, which is our lead product, in the clinic, LN-145, which will enter the clinic in the next few months. We have all of our licenses from the NCI, and we continue with our CRADA, working with Dr. Stephen Rosenberg. Thank you very much.
Great. So for 144, maybe you can give us an update on enrollment timelines and when we're likely to see the data emerge.
Right, so the important timeline for us is to have an end of phase 2 conference with the FDA. It's really important as we move into phase 3 next year, and that is certainly our goal. We would like to have an end of phase 2 with FDA by the end of the year to discuss both clinical and CMC related issues.
Are we on track to see data at ASCO or an interim look?
We'll probably do just after ASCO.
Just after?
Just after ASCO, yes.
What might we expect to see at that just after ASCO, and what would the forum be to see?
Too early to say.
Okay. Maybe you can quickly outline as Lion Bio's taken over the program, what's involved? What is the patient experience? What do they go through in the LN-144 protocol? And how might that ultimately differ in the real-world application pending phase 3?
So from a patient perspective, the patient has a small surgical event. So we take a biopsy or a piece of tumor that is about the size of a sugar cube. At that point, the patient goes home. And while the patient is home, we are making TIL for this patient. The process involves what is known as a pre-rep part of the process, and then rep, which stands for rapid expansion protocol. Now, some patients' TIL grow much faster than others, and so the total time for some patients would be quite short, and for others it would be probably combined in the 4.5-5 week time frame. A week before the patient is administered TIL, they come back to the hospital where they are lymphodepleted with Cytoxan and fludarabine. It is not an in-hospital stay. They're only there for one night. The rest of the lymphodepletion is done at home so they can go home. Then they get hospitalized again on the day of TIL infusion. And it really is not for the TIL infusion, but for the IL-2 that the patient gets afterwards. Patient only gets 5 to 6 doses of IL-2, which equates to about a day or a day and a half. They may stay in the hospital for a little bit longer because we need to wait for the fluid accumulation to resolve itself, and then the patient goes home, and that is it.
Got it. And that would also reflect the real world experience?
Absolutely,
Yeah.
absolutely. I cannot see the difference between what we do in the clinic now versus in the real world.
And I think you have some data in patients who've previously been exposed to checkpoint inhibitors. Maybe you can summarize what you're seeing in that setting.
Yeah, so this is data from the NCI again. So Dr. Rosenberg has on numerous occasions at SITC and also at ACR mentioned that the outcome of TIL is not really all that dependent on prior therapies. I do remember seeing some data on previously treated ipilimumab patients. And if memory serves me right, there was a cohort of about 30 patients or so, and there certainly didn't look like much of a difference in outcome in those patients. The cohort of patients who had received anti-PD-1 antibody treatment is very tiny. So I do want to caution you in terms of making too much of that data. And again, there it is also Merck murky because some patients have only received PD-1 and others have received a combination of PD-1 and ipilimumab. But again, my expectation is that it is probably going to be a little bit less than patients who had not seen checkpoints, but not devastatingly so. So I'm expecting something in the 40% range or so.
You have a recent collaboration with Medimmune. Maybe you can elaborate on the parameters of that.
Absolutely. So, so the idea with Medimmune, so the arrangement is actually twofold. There's a preclinical component and a clinical component. The clinical is to study TIL with Durva, which is Medimmune's anti-PD-L1 antibody. And there's a lot of preclinical reasons for doing that. We have some preclinical data that shows there's a potential synergistic effect. From a preclinical perspective, it's really quite interesting because we have an opportunity to help Medimmune understand some mechanism of action for their antibodies. And the benefit to us is that we can see what those antibodies do in our manufacturing system. And so it is really, truly a win-win arrangement.
And the phase 2 that you have ongoing, is that going to be all comers? Will it be primarily checkpoint inhibitor experienced patients? And how ultimately does this set you up for the end of phase 2 meeting with the FDA?
This is the phase 2 for what?
I'm sorry, 144.
Oh, 144, yes. So for 144, it is patients who have failed checkpoints. So they must have failed at least one biological treatment, and so most of these patients will have had anti-PD-1 and or IPI treatment. And that is certainly where we are thinking about positioning TIL on the market, and also obviously for phase 3 or pivotal phase 2.
And how many patients in this phase 2 are you expecting?
Well, we had said that we won't go over 20 patients, but that is completely an arbitrary number.
And what's your sense of what might be required for approval based on precedents in the checkpoint inhibitor refractory setting?
So that is $1 million question, but certainly what we would like to petition the FDA with is a single-arm study in patients who have failed checkpoints. And so for them, there really is no other approved treatment. And I think one can probably do that with somewhere between 50 and 100 patients.
A number of different things you can do with the TILs. You can gene edit them, and you can try to knock out PD-1. You can, I guess we've talked a little bit about the different opportunities. What are you most excited about in terms of next gen, and maybe also including, you can replace the IL-2. You can try something a little bit easier, gentler. Of all those options, where do you kind of see the platform going?
There are just so many opportunities here. From the lymphodepletion, which I think one eventually, perhaps post commercialization, one can simplify that dramatically. There are also additional IL-2 analogs in development that could be very interesting for us, such as pivotals, IL-2, or alchemy, and so forth. In terms of modifying the TIL themselves, I think there are lots and lots of opportunities. We can, for example, change the tumor microenvironment or have influence on the tumor microenvironment by pulling in more T cells. And you can do that with a TIL-like receptor pre-treatment, and then harvest those TIL and see how they grow. Something like that is very, very high on my agenda. But I think probably the most exciting opportunity would be in a gene-edited TIL, where we could knock out the appropriate genes, whether it's PD-1 or TIM3 or whatever the case may be. So definitely stay tuned on that.
Great. Well, I think we're out of time.